phone it over here, Lauren?
Yes, sir. I just need to put this in the...
Oh. Seems like it's pointing down here. You can see that too?
No. I have to hold it to the screen.
Okay, this guy Mesher, this is chapter 14. We're studying DNA, nucleic acids. And this guy is the guy who discovered nucleic acids. He took white blood cells and somehow chemically broke down the cells so that the nucleic acids came out of the nucleus. He called them nucleic acids because they came from the nucleus. Isn't that a good name?
Yeah.
It's really good.
I don't know how he did it, but he did it. And there's a famous experiment called the Griffith transformation experiment that kind of helped prove that nucleic acids carry information. And this one's not easy to follow, so you're going to have to hang with me here. Now, this involved mice. This experiment involved mice and certain types of bacteria. Now, there's a certain type of bacteria that has a kind of a capsule on it, a smooth capsule surrounding the bacteria. Griffith called these S-strain bacteria. S meaning it's got a smooth capsule surrounding the bacteria. If you inject mice with S-strain bacteria, it'll kill the mouse. It's a deadly infection. Thomas Bill, are you with me here?
I'm looking at the mice.
Okay. So, get this in your head. S-strain bacteria kill mice. Now, there's a different strain that is basically the same bacteria, but it doesn't have the capsule. He called them R-strain bacteria because they look rough on the outside. They don't have the smooth capsule. Cartier, are you with me? Seems like you're staring at the spot. If you inject mice with R-strain bacteria, the mice survive. So the S-strain bacteria kill the mice, the R-strain bacteria don't kill the mice. Okay? Now, this is where the experiment gets interesting. If you take S-strain bacteria and you heat up the test tube that they're in so hot that it kills them, if you inject the mice with the heat-killed S-strain bacteria, it doesn't kill the mice because the bacteria are dead. And dead bacteria can't multiply and kill your mice. It won't cause an infection. Are you with me so far?
Mm-hmm.
And here's the capper. What if you heat kill the S-strain bacteria, put them together in the same hypodermic needle with R-strain bacteria and inject both of those into the mouse? Now remember, R-strain bacteria didn't kill the mouse, and heat-killed S-strain bacteria didn't kill the mouse. But if you put the two together in the mice, it dies. Furthermore, if you take a blood sample from the dead mouse, there's living S-strain bacteria in the blood sample. How could this be? How could it be that you put dead S-strain bacteria into the mouse's body with live R-strain bacteria, and then you withdraw live S strain bacteria from the mouse. Yes, Meryl.
It mutated it. Mutated the R strain.
What did?
The R strain.
S strain did.
Right.
But you killed S strain.
The R strain used genetic material from the S strain to make copies.
That's what they concluded. So what they concluded was the R strain bacteria must have picked up genetic information from the S strain, the dead S strain, and used that genetic information to make the smooth coats, which ends up killing the mouse. And that's why they could withdraw S strain bacteria. In other words, the R strain bacteria got transformed into the S strain. That's why this is called the Griffith transformation experiment. And it was the first experiment that showed, that proved that molecules, some sort of molecules, must have moved from one bacteria to another and changed that bacteria. It's a very famous experiment. Griffith won the Nobel Prize for it. Some molecule can change the organism. That's big news. Now, what molecule was it? They didn't know at the time. Was it DNA? Was it glucose? Was it some kind of protein? Was it some kind of lipid? They didn't know what it was. So that's what they set out to kind of figure out. Are y'all happy about the Griffith transformation experiment?
Yes.
Do you even care? Look at the nice living mouse. Look at the poor dead mouse.
It's not a big deal.
I used to have at
Scientists kill a lot of mice in their research. But it leads us to figure things out and be able to do things like make vaccines, which can save lives. Here's another famous experiment you have to know about. First of all, does anyone have any questions about the Griffith transformation experiment?
Was this before the other dude found nucleic acid?
Good question. Does it say
Or
the
did
date?
it not like correlate?
Does it say the date that
This was after.
this was in the 1900s, was it?
It's 1928.
1928, so it's well after.
Okay. So you're just gonna-
So he discovered nucleic acids, didn't know what they really did. And then Griffith did this experiment that showed that there was some kind of transforming molecule that was passed along. Now, here's another famous experiment that showed that a transforming molecule is a nucleic acid. So this is this is another experiment where we really learned something. And it is called the fly experiment. I can't remember the name of this. Does it say in your book? Anyone have the book open? This is
Hershey and Chase.
Hershey and Chase, yes, the Hershey and Chase experiment. Hershey, like Hershey's chocolate. Don't know if he was related to her actual Hershey chocolate version.
That'd be kind of cool.
The Hershey Chase experiment. Are you ready to get in pay attention mode?
Yes.
So you can understand this one? This one's not easy to understand either. They know viruses cause cells to be infected and make new viruses. But Hershey and Chase wanted to figure out what was it that gets passed into a cell from a virus. And so they studied viruses And they figured out viruses are really only made of two things, at least the ones that they were working with. Viruses have a protein coat that kind of looks like this. This is protein. And then they have nucleic acid on the inside. And so they were wondering, when a virus infects a cell, does the protein go into the cell and tell the cell to make more viruses? Or does the nucleic acid go into the cell and tell the cell to make more viruses? They didn't know. They didn't experiment to figure it out.
Was it RNA?
The nucleic acid can be either DNA or RNA. In the coronavirus, it's RNA.
Yeah.
In chickenpox, it's DNA. It depends on the virus. In the viruses they were using DNA viruses.
Is it easier to mutate if it's RNA?
It's easier to mutate if it's RNA. That's why you need an annual flu vaccine, and why we may end up needing an annual coronavirus vaccine. We may end up having to get that because RNA does mutate very easily. The one thing we do have with the coronavirus over the flu is the coronavirus just evolved last year. If everybody gets the vaccine, we may be able to stomp it out before it can mutate enough to be different. We didn't do that when the flu first started, however many hundreds of years ago, and now there's so many different flu viruses out there that we can't get vaccinated for all of them. And so the flu's going to come around every year. We may be able to stop this coronavirus. We don't know. It's an interesting idea. But there's a lot of people out there who don't want to get the vaccine. And if a significant percentage of the population doesn't get vaccinated, we'll never stop the coronavirus now.
Isn't it like 40?
It'll continue to live in those people that don't get vaccinated.
Why do they want
And it'll mutate and come back at everyone, and we'll have to get shots every year. So not only should you get vaccinated, you should talk people into it who say they don't want to get vaccinated. Because if we all get vaccinated, it can knock the whole thing out like we did smallpox. I don't know if you remember, but smallpox was a terrible disease. We vaccinated everyone in the world against it, and it disappeared. There's no more smallpox.
Smallpox is that it's DNA. It took them like 20-something years
It's a
to
DNA
vaccinate
virus.
everybody.
It's a DNA virus.
And if it takes 20 years to vaccinate this, it'll mutate.
We got to vaccinate everyone
In a year.
A year or two. Yeah. What was the question? Okay. Well, so let's see the experiment. Sorry, I got off on a tangent, but it's nice to talk about coronavirus because it's going on right now. Anyway, so this virus would land on a cell. They infected E. coli cells with it. And so what did it inject? Here's how they figured it out. It was an ingenious experiment. Protein has sulfur as part of its buildup. Do you know where the sulfur is in protein?
It's the amino acid.
It's part of it. Proteins are made up of amino acids.
Disulfide bond.
And an amino acid, do you remember an amino acid looks like this?
Is it cysteine?
Hold on. Do y'all remember this is what an amino acid looks like?
Yeah.
Where's sulfur? I don't see any sulfur.
It's in one of the R groups.
It's in one of the R groups of one of the amino acids. Cysteine. Cysteine has sulfur in it. And these scientists knew that protein has sulfur. Nucleic acid doesn't have any sulfur in it. So what the scientists did was they radioactively labeled the sulfur in the protein. How did they radioactively label it? I don't know, but they did. And so the protein, as you see here, they have it red. The protein is radioactively labeled with sulfur 35. That's a radioactive form of sulfur. And what it basically does is it makes the protein glow. That was one group. Now another group, they took viruses. I'll draw them over here. I like drawing viruses. Very cute. And instead of labeling so in this group, I'm gonna use orange for the radiation. In this group, they radioactively labeled the protein with S35. In this group, they radioactively labeled the nucleic acid with P32, which is a radioactive form of phosphorus. They basically, what they did, I do know how they did it, they grew viruses in cells that only had over here, that only had S35 as a type of sulfur. And so all these viruses that grew had to use that S35 sulfur. So now their proteins glow. In these viruses, there was only P32 in the cells, and so they had to pull that in to their DNA, and so their DNA glowed. And so what happened was, they then just infected the E. coli cell with these two groups. And then, so here's this radioactive phosphorus labeled cell, virus, injects its DNA. We know now that the DNA is what gets injected. Question?
Is S35 the only one that glows?
S35, the sulfur glows.
Yeah.
P32, the phosphorus glows.
Phosphorus?
So nucleic acid has phosphorus in it, and protein doesn't. There's no phosphorus in protein, but there's phosphorus in nucleic acid. So the nucleic acid has radioactive phosphorus. It's a good question. I should have said that. So the radioactive phosphorus is glowing here. The radioactive sulfur is glowing here. And so what happened was they infected the cells with the two radioactively labeled viruses. And they put them in a test tube and spun them around. And the heavy cells went to the bottom. And the light viral coats stayed at the top. Y'all know how a centrifuge works? You ever seen that? It spins around really fast, and heavy things go to the bottom, and the light things kind of stay higher up. So they could separate out. After the viruses landed on the cells and infected the cells, then they spin them around, and these little virus coats come off of the cells, you see, and float up to the top, and the heavy cells go down to the bottom. And they look for where the radiation was. So in this group out here that has radioactively labeled phosphorus, all the radiation was down in the cells. Meaning that they had injected DNA into the cells, not protein. If protein had been injected into the cells, then, and DNA not injected into the cells, then it wouldn't be radioactively, wouldn't be glowing down here. And the same thing right here. When they infected cells with radioactively labeled protein, and then they spun them up in a centrifuge, these cells didn't glow radioactive because the radioactive protein stayed on the outside of the cells. You see, this stuff is too small to see. You can't actually see viruses, even with microscopes. They didn't have electron microscopes at the time. So you can't see what's being injected into the cell. You have to figure it out through experiment. And they knew, now after doing this experiment, that DNA gets injected into cells and protein stays outside of the cells. And the cells, once they get that DNA, make more copies, so it had to be information in the DNA being passed into the cell. DNA must carry information, not protein, to tell cells what to do. Big experiment. Won them the Nobel Prize. You know what you get if you win the Nobel Prize nowadays?
A lot of money.
Money.
$1 million. Did that make sense, or did I botch that trying to explain it?
Makes sense.
Since he made
Ask me if you got lost somewhere. Did y'all, you want to see video footage? Would that help?
Yeah.
What was the point of doing it again?
To figure out if it was protein that was being injected into the cell, or if it was nucleic acid that was being injected into the cell.
I'll take a break.
Did that make sense, Charlie?
Yeah.
To see if it was protein injected or if it was... Okay, let's just see video footage, see if this helps.
In the Hershey Chase experiment, bacterial viruses called phage were used to demonstrate that DNA material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing P32 labeled deoxyribonucleotides. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
See, they can follow the radiation. If the radiation is in the cells, when they radioactively label the DNA, then they know that the DNA must have passed into the cell. Thomas, you got a question? That's the Hershey Chase experiment. Another experiment you'll hear about, but it comes a little later. Aha. Was this X-ray diffraction experiment. Now, this actually comes in tomorrow night's reading, but I'll go ahead and tell you about it now because it's in experiments, and they'll ask questions about experiments. So, there's a lady, her name was Rosalind Franklin, who did this experiment where she fired, once they found out that DNA is the thing that carries the information, they wanted to figure out what did it look like? What does a DNA molecule look like? It's too small to see. How could they figure that out? Well, one thing that can penetrate small molecules is X-rays. X-rays have a much shorter wavelength, and so they can kind of get into cracks and crevices between molecules. Visual light, like we see with, has really long wavelengths and has and the rays of it, I guess you could say, can't get too tightly packed together. And so they can't really penetrate and find out any information about really small molecules. In other words, there's no way to see with eyes very small things. But you can see it with X-rays. So this lady, Rosalind Franklin, took DNA and she froze it where it formed crystals, and she shot X-rays into it. And the X-rays would bounce off of the DNA and make patterns. It's a whole science to it. It's called X-ray crystallography. And she was an expert in it. And she produced this. This is one of the pictures she produced. Now, you and me look at that. Just looks like a kind of a cool pattern. But if you know how to read X-ray crystallography, you can make some sense of that. And one thing that tells you is that DNA is a helix shape. It's shaped like a corkscrew. And so she took that picture, and that shed some light on the shape of DNA. And then these two guys, or these three guys, the main two guys were Watson and Crick, and there's a third guy, McCarty. And I think that's Watson, and that's Crick, and that's McCarty. And they came up with and built a model of DNA based on what they knew about it, using in part Rosalind Franklin's picture. And that's the model of DNA that we have. And only recently, this is kind of cool, in the last 10 years, have they developed microscopes that could see the structure of DNA as they had come up with. They said DNA is a double helix structure that looks like this. And they got the Nobel Prize for figuring that out. Rosalind Franklin didn't get a Nobel Prize because she was dead already. And they don't award it to dead people.
Right.
And also, some people think that because she was a woman, people didn't want to give her the credit she deserved. But anyway, that's the DNA molecule when they figured out what it looks like. And in the last 10 years, they've actually developed microscopes that can see it, and it does look just like they said it would. And so it finally confirmed, without any doubt, the structure of DNA. So tomorrow, what we'll do is we'll go through the DNA molecule and look at it, and we'll see exactly how it works. We've already talked about this a little bit earlier in the chapter. So, should have a quiz on 14.1. Peace.